Welcome to The Explainer. Today, we are on the trail of a molecule that holds the secrets to, well, you. We're talking about DNA. We're going to follow the clues from a huge, century-old scientific mystery all the way to a modern crime scene to figure out how the blueprint of life was finally revealed.
So, let's start at a crime scene. Imagine this. A forensic team finds just one tiny, almost invisible hair. And from that single strand, they can build a genetic profile so ridiculously precise that it can point to a suspect with almost perfect certainty. I mean, how is that even possible? How can something so small solve such a big crime?
Well, the answer is something called a DNA fingerprint. You can think of it like a genetic barcode that's unique to pretty much every single person on the planet. And you can see right here, there's a clear match between the evidence and suspect too. But to really get how this works, we have to rewind. We've got to follow the trail of clues that scientists spent decades uncovering.
Okay, so for the longest time, scientists knew that, traits were passed down from parents to children. That part was obvious. But what was carrying that information? What was the messenger? That was the big mystery. For a while, the prime suspect was protein. I mean, it made sense, right? Proteins are super complex, they do all sorts of things. But there was this other, much simpler molecule that everyone was kind of overlooking, and it was about to step right into the spotlight and just change, well, everything.
So the first half of the 20th century, man, it was just a whirlwind of discovery. It was like a scientific sprint. The trail really starts in 1928 with this mysterious thing Griffith called a transforming principle. Then, by 1944, a team led by Avery points the finger squarely at DNA. They've got their prime suspect. Fast forward to 52, and the Hershey-Chase experiment comes along and provides the smoking gun. And then just 1 year later, boom, the final piece of the puzzle clicks into place when its iconic structure is revealed.
But why was there even a debate? Well, you have to put yourself in their shoes. On one side, you had proteins. They're these super complex molecules, built from 20 different amino acids. They just seem like the obvious candidate to carry all of life's complicated instructions. And then on the other side, you had DNA. It's built from just 4 simple bases. 4. To most scientists at the time, it just seemed way too simple for a job that important. It just didn't seem possible. And this is where a brilliantly simple experiment from 1952 comes in. Seriously, it's so clever. Alfred Hershey and Martha Chase used viruses that infect bacteria. They made 2 batches of these viruses. In one, they tagged the protein coats with radioactive sulfur. In the other, they tagged the DNA cores with radioactive phosphorus. Then, they let these viruses infect the bacteria. And here's the best part. They used a kitchen blender to literally shake the viruses off the outside of the bacteria. And the result? The radioactive DNA was found inside the bacteria, while the protein was left outside. That was it. That was the proof that DNA was the stuff being injected.
So just 1 year after that killer experiment, the final secret was unlocked. Using this absolutely crucial X-ray image, it was called Photo 51, an image captured by the brilliant scientist Rosalind Franklin, who for a long time didn't get the credit she deserved. Well, using her data, James Watson and Francis Crick finally pieced together the 3D puzzle of DNA's structure. And what they found was that iconic, elegant shape we all know today, the double helix.
Okay, so we've got the blueprint. Awesome. But think about it. A cell has thousands and thousands of genes. It's not going to use all of them at the same time. That would be chaos. It's like having a giant cookbook. You don't cook every recipe at once. So the big question is, how does a cell know which parts of the blueprint to read and, just as importantly, when to read them?
Well, in bacteria, the solution is actually super efficient. They group genes that are all related to the same job together. These units are called operons. The best way to think about it is like a power strip for your tools. You plug in your drill, your sander, your saw, but you only have one master switch to turn them all on and off at the same time. It's a brilliant way to coordinate a whole team of genes to get one specific job done.
Okay, a classic example of this is the lac operon in E. coli bacteria. So if there's no lactose around, that's the sugar in milk, it would be a total waste of energy for the bacteria to make the enzymes to digest it, right? So what happens is a little protein called a repressor physically sits on the DNA right in front of those genes. It's like a roadblock. It blocks them from being read. The switch is firmly in the off position. But what happens when lactose does show up? Say you drink a glass of milk. The lactose molecules bind to that repressor protein, and that changes its shape, making it fall right off the DNA. And just like that, the roadblock is gone. The path is clear. The cell's machinery can zoom in, read the genes, and start making the enzymes it needs to digest that sugar.
This is a really important idea. The environment around the cell can directly flip its genetic switches on and off.
Okay, so let's bring it back to the crime scene. We know DNA is the blueprint, and we know it has on and off switches. That's great, but how in the world do we get from that one tiny microscopic hair follicle to that big, clear genetic barcode we saw at the beginning? To do that, we need a modern scientific toolbox, and inside, there are two absolutely essential tools.
Tool number one is something called PCR. That stands for polymerase chain reaction. And honestly, the best way to think of it is as a genetic photocopier, but like on an epic scale. It lets scientists take one tiny, basically unusable speck of DNA and just amplify it, making billions and billions of identical copies. Now that is enough to work with and create a clear profile.
So how does this genetic copy machine work? Well, it's all about a cycle of very precise temperature changes. Step 1, you heat the sample up to about 95 degrees Celsius. It's really hot, and it causes the DNA double helix to unzip into two separate strands. Step 2, you cool it down. This lets these little guide molecules called primers find and stick to the exact spot on the DNA you want to copy. Step 3, you heat it up a little bit again, and a special enzyme called Taq polymerase gets to work, building a new copy of the DNA, starting right where those primers land.
And here's the magic. You don't just do that cycle once. You repeat it again and again, 20, 30, sometimes 40 times. And every single cycle, you double the amount of DNA. It's an exponential explosion. One molecule becomes 2, 2 become 4, 4 become 8, and in just a couple of hours, that single starting molecule becomes billions. Now we finally have enough evidence to analyze. Okay, great. We have billions of copies of our DNA evidence. But how do you sort through all of that to create that unique fingerprint? For that, we need tool number 2, gel electrophoresis. And the way it works is so cool, because it all comes down to one very simple, basic fact about DNA.
So, here's the thing about DNA. Because of these things called phosphate groups in its backbone, the whole molecule has a negative electrical charge. And what do we know about charges? Opposites attract, right? So if you put that negatively charged DNA into a gel and run an electric current through it, it's naturally going to start moving away from the negative side and toward the positive side. Simple as that. But here is the really ingenious part. The gel isn't just empty space. It's like a thick molecular mesh, basically an obstacle course. And just like in a real race, the smaller, shorter fragments of DNA can wiggle through that mesh really easily. They move farther, faster. But the big, long fragments, they get tangled up. They move much more slowly. And just like that, this simple process perfectly sorts all of those billions of DNA fragments by their size.
All right, let's go back to our crime scene for the big reveal. We've used our genetic photocopier, PCR, to make billions of copies of the DNA evidence. And we have our molecular sorting machine, gel electrophoresis, all warmed up and ready to go.
So we load up the DNA from the evidence, and we load up the DNA from our suspects, and we run them all through the gel. And what do we see? Those bands. Each band is a collection of DNA fragments all sorted perfectly by size, creating a pattern that's totally unique, the DNA fingerprint. And you can see it clear as day. The pattern from the evidence is a 100% match for suspect 2. And just like that, the case is solved.
Look, DNA fingerprinting is an absolutely incredible application of our ability to read the genetic code. But in the last few decades, science has taken another, even bigger leap forward. So what happens when we learn not just to read the code, but to actually rewrite it?
Well, that brings us into the world of recombinant DNA. Scientists have discovered these amazing molecular scissors, they're actually enzymes, that can cut DNA at very specific places. This allows them to literally cut a gene out of one organism's DNA and paste it into another's. It's basically a biological copy and paste.
And the applications of this? They are already changing the world. For example, we can take the human gene for insulin, paste it into bacteria, and turn those bacteria into tiny living factories that pump out the insulin needed by millions of people with diabetes. It's incredible. And it's not just insulin. This same tech is used to create human growth hormone, to develop gene therapies for terrible genetic disorders, and even to engineer crops that are more resilient to help feed our planet.
So think about this journey. We went from a fundamental question about what makes us who we are, to a tool that can solve crimes and ensure justice, all the way to a technology that can literally rewrite medicine. Our understanding of DNA hasn't just changed science, it has totally transformed our world.
We can now read, copy, and even write the code of life itself. The power in our hands is just immense. And the next chapter in this incredible story, well, that's a chapter that we are all going to write together.